Okay, so starting, as I've told you just a minute ago, we're recording this, and the second thing I've told you is that you have all the work that was due today is going to be due on Monday, so you have it with you to study for your test. You have a 20-question test on cells and cell types on Monday. Is that clear?
Yes.
All right. So, classification of cells. One of the things you have to, first of all, let's talk about this again, that your work and your notes all together are feeding into this test. So the notes on this video and the work you've done, that's where I'm getting the questions on this exam. Okay. So when we're looking at cells, this is not a concept map. This is an organizational chart. So in a way, it's a subtype of a concept map. And the only reason I'm saying this is because some people ask, why is single-celled not connected to prokaryote? Right? Because it's not a concept map. The objective here is that you understand that all organisms fall into one of these categories. All organisms fall into one of these categories. Okay, so in your organizational chart, you see an organism, everything that's alive, everything that's an organism falls in this chart. And we'll talk about cell theory after Monday. So a prokaryote is a cell, is a living creature that number one is, let me try to change the color here. Number one, a prokaryote is single cell. Number two, it has no nucleus. You all should know that by now, right?
Yeah.
Okay. Number three, it has, they can be one of two types. It can be a bacteria. So bacteria, or it could be what's called an RK bacteria. Those are the two types of single cell organisms, prokaryotes. Are we good with that? So an archaebacteria is just a type of prokaryote. We'll go into it more deeply later. It's it was discovered in the 60s, in the mid-60s. Before then, we thought all bacteria were the same. What we found is that some bacteria can live in really extreme environments like salt marshes and hot water springs where water is near boiling, but they're able to survive those areas. We find that these bacteria are what we call extremophiles. They actually exist in places where we would assume life would not exist. Highly acidic, highly basic, high temperatures, extremely low temperatures.
So is it bacteria that exist?
It's bacteria that evolved. They've evolved into a separate group. And so we'll talk more about them later. But I don't want you to think that it's just bacteria. There's actually very little difference between bacteria and archaebacteria when you're just looking at them. Okay? So their genes, their proteins, some of their internal structures are different. But in general, for sure, they're both what?
Bacteria.
They're both what? Prokaryote. Archaebacteria and bacteria are two groups here, but they're both prokaryotes. They're both single-celled. They both have no nucleus. Okay? So this is two major groups of life. So the other major group of life are these things called eukaryotes. We are eukaryotes. Eukaryotes are simply, and I know you know this already, they're what? Okay. But what makes a eukaryote different from a prokaryote? Thank you. It has a nucleus. Has a nucleus. Now, you might be tempted to say, well, they have more than one cell. They're multicellular. Look at the organizational chart, though. Are all eukaryotes multicellular? No, they're not. Some are single-celled. And what are some of those examples? There are these things called protists. Protists are things that are, an example would be ciliates. You might think of something that's an amoeba. All right? So these organisms are single-celled, but what do they have? A nucleus. Algae, yes, that's That's the major characteristic difference between multicellular and single-celled eukaryotes. Prokaryotes? Yeah, the difference between prokaryotes and eukaryotes, remember what I said to you earlier when we talked about membranes, is that there's one more thing about bacteria, prokaryotes. They have no membrane-bound organelles. So all those organelles that you learned about over the weekend, right? All the lysosomes, the endoplasmic reticulum, the Golgi body, the nucleus, the nucleolus. These organisms don't have any of that. They just have the outer membrane, and then in the middle they have this little nucleoid region, and that's it. So you need to remember that these two groups of life, bacteria and archaebacteria, they have no membrane-bound organelles. They function, but we talked about this. Remember when we talked about the difference between a house and a shack? They do everything in one room. All right, where eukaryotes divide things into different rooms. Right, and of course they need membranes to do that. So that's single-celled, and that's Protists. But they also there's some single-celled fungi. Do you know what fungi are?
Yeah.
They're funguses, the plural funguses, fungi. And there's one type of fungus specifically that comes to mind is called the yeast. So everybody should be familiar with yeast. We make our beers with yeast, right? We make bread rise with yeast, right? You can get a yeast infection, right? Both males and females, by the way, ladies and gentlemen. So yeast can actually, and yeast infections can happen in dogs' ears. So these things are fungi. They grow in wherever there's moist and dark. Yeah.
Do they speed up the reaction? Like some yeast?
Oh, how do yeast help how do yeast make bread rise? Is that what you're asking?
Not just that,
How they do what they do, we'll talk about it later. They produce carbon dioxide. That's how they help bread rise. They also produce alcohol. When they digest sugars, they can produce alcohol through anaerobic respiration. But we'll talk about that when we start to do respiration and photosynthesis, which will be probably after Thanksgiving break. Because during Thanksgiving break, we're going to be talking about mitosis and meiosis. Are we all good? All right. So when we're looking at multicellular organisms, there's two major classifications of multicellular organisms, and that's plants and animals. Now this is simplified. There's all kinds of little nuances that we're not really going to get into right now. But what you have to know that the test is what you see right in front of you right now. Is that clear? So you need to know this. Somehow you have to get into your brain to be able to answer questions. All right. This is kind of a review of what you did over the break, right? You had eukaryotic cells, animals and plants and bacteria. I numbered, obviously archaebacteria and bacteria, right? So that's why I added archaebacteria and I misspelled it, but that's fine. So I'm just going to have to, I wish I could delete that, but I can't.
So,
I can't, maybe I can, yeah, good.
prokaryotes got ribosomes?
Huh?
Prokaryotes got ribosomes?
Yes, ribosomes, bacteria do have ribosomes. They're actually different from eukaryotic ribosomes. They're smaller, but they're ribosomes. So that's one of the organelles they have. People tend to leave them off. Why do you think they have ribosomes and none of the other organelles? They're not membrane bound. These ribosomes don't have membranes. They're just RNA. We'll talk more about that when we do DNA and RNA. We're all good with this? Okay, so now let's go on. So these differences, I'm not going to go over them. You did activities that separated your plant animal cells, right? And everybody now, the only thing you have to add now is this, right? And of course you should know Protists are single-celled what? Eukaryotes. So there's 4 groups of eukaryotes, right? At least 4 groups of eukaryotes. There's animal, plant, Protists, which are single-celled, right? And fungi, which is single-celled and multicellular, right? So it depends on the life cycle, etc. All right, are we all good? Yeah.
What would they call a mushroom? Because a mushroom and fungi, so would it be?
That's a good question. That's one of those nuanced things, right? So multicellular, sometimes single cell. When it's a spore, when it's growing, there's things like slime molds that form multicellular organisms in the forest floor. They actually move towards each other. It's kind of creepy. We can show some video later on it if you like. But then you're standing in the woods and all of a sudden you see the leaves rustling and they come together and they form almost looks like a mushroom. It's like a slimy though mushroom, in different colors. And then they split up and they go off about their business. So it's really, they're single cell turning into multicellular, going back into single cellular. So it's kind of crazy. Life is crazy. There's all kinds of different exceptions. Yeah?
That's really how it is.
Yeah, that's really, I'll show you the video if you like, yeah. They're called slime molds. They're very interesting. But in any case, the point is you have these different cell types. All right. So, let me get rid of some of this older stuff. Hopefully it doesn't, yeah, good. And when we're talking about humans, as you know, we are eukaryotes, right? Nobody has a problem with that?
Yeah.
Because I don't need you to have a problem with not understanding that we are eukaryotes. I'll just leave the rest there, I guess. All right, so some of the basic, this is just some of the very different cell types you have. And one of the things you have to consider when you're talking about cell types is this: structure influences function. You can even say determines. And vice versa. They influence each other. They determine each other. So, see, I hate when it sticks to it. So the structure influences the function. Okay? So just like we talked about with doors and windows, right? A door has to open, otherwise it's a wall, it's not a door. A window you have to be able to see through it, otherwise it's not a window, it's a wall, right? So, you look at red blood cells, what are the function of red blood cells? What do they do? Give me a guess.
Give us instructions? No, wait, that's
No.
what it is. My bad.
A red blood cell, what does it do?
They keep the actual blood liquid.
No?
Carry
They carry oxygen, right?
I'm so wrong.
And so they're carrying oxygen, they're in the blood, right? They're in the blood, so that means that they kind of flow in every nook and cranny, right? They're in these little tubes, the tiny little tubes called capillaries. So they have to flow smoothly. So let me ask you something. Let's think about in life where we might have something flowing down, let's call it a river. Who's been to Kalahari or water parks in general? Anybody? Who's gotten in one of those river rides? What's the shape of it's this kind of inner tube shape, isn't it? Why do the donut thingy, why do they look like little donuts?
They carry you.
Because because they don't get stuck on the corners, so that when the river makes a bend, you don't get them sticking to each other and making blocks. They just kind of bounce off and keep turning, right?
Yeah.
And that's why red blood cells have their shape, because they got to squeeze on through tiny little spaces called capillaries, and you don't want them getting stuck. In fact, interestingly, we'll talk more about this later, sickle cell anemia is when the cell, instead of being round, looks like that. And so what happens to that cell when it gets to the capillaries? They get caught, they get stuck. It causes pain and blockages and bleeding. It causes a lot of problems. So sickle cell anemia, so you can see that when I hope you understand that this idea of structure and function being linked, this is a perfect example. In fact, we're going to use sickle cell throughout the year as a really great example of a lot of biological functions. And one of them, structure and function being linked, is a key part. So red blood cells have their shape because of what they do. Quick questions, we don't have a lot of time.
So what happens when you have a trait? Does it still circle?
So you have a what?
Sickle cell trait.
More complicated, let's write that question down. When we cover sickle cell anemia in whole, and we'll have a whole case study probably next week, Thursday. Ask that question again. Yeah.
So, what about when your blood clots?
A regular blood clot or a blood clot because of sickle cell?
Great.
Oh, regular.
So blood clots, what happens is when you do damage to the blood cell, and I don't want to go into this in detail, they releases certain proteins that make what I want to, let's just call it a Band-Aid. And they stick to each other, and they stick to the side of the blood vessel. That stops bleeding. You want blood to clot when you're bleeding so you don't bleed to death, right?
Right.
Now the problem comes in that when that blood clot dislodges and starts to flow like a red blood cell through the bloodstream, that blood clot can get stuck in capillaries, and those capillaries can be in places like your brain or your heart or your lungs. And they caught in your brain and they stop blood flow in your brain, we call that a stroke. If that blood clot gets stuck in your heart and stops blood flow, we call it a heart attack. And if that blood clot gets stuck in your lungs, we call them pulmonary embolisms. So it's that simple idea, when you just asked, that simple idea of a blood clot leads to three major issues in the human condition. Is that interesting? All right. So, anyways, the point of this slide is to make you let you see that not all cells have the same shape. They're not all the same, expressing the same proteins. Now, what's a major thing that you must know for end of course exam, for any understanding to go on, for anything is this: all of these have the same DNA. So you have all of these cells. Well, all of them except the females don't have the sperm and the males don't have the ovum, right? You should know the word ovum means is your egg cell. Another word for egg cells. That's why we call your ovaries ovaries, right? We call your ovum, the egg, an ovum. And when you release an egg, we call it ovulation. So it's all based on the same root word, okay? But obviously the females have this and the males have this, right? So that shouldn't be a surprise to anyone in this room. If it is, okay, we'll talk about it later. All right, so all of these have the same DNA. So it's interesting that you can have red blood cells and these epithelial cells, they all have the same DNA. So how come they're different?
Functions.
But exactly, they all have different functions. But their structures are different, their functions are different, that's why they have different components. But how come they, remember, what did you learn over the weekend? The DNA commands the cell. So how is it that you can have the same DNA in the red blood cell, epithelial cell, smooth muscle, bone, nerve, and sperm, and or ovum, right, depending on male or female? How do you have the same DNA in each of these, and they have such radically different structures and functions? Okay, they're also right, because they're you. Because every cell that makes you up, all trillion cells, all started with one single cell. So all of your DNA is the same. But how is it you have an eye forming an eye and ears an ear if the DNA is the same? The instructions that make you are in the DNA. How is it that you have these different cell types, these different tissues, if all the DNA is the same?
The different organelles.
No, the organelles are all the same, except for a couple exceptions.
Our genes.
What about your genes?
The traits.
So you have your DNA, and your genes are here, but all the genes in each of these is the same for every single human being. For you yourself, your chromosomes, your genes, they're all the same. So how is it that your eye can be different from your nose, from your lips, from your muscles, from your nerves?
The RNA.
No, it's good guess.
What'd you say?
But not quite. I mean, yes and no is your answer to that one. That was a good, that's a really good guess.
Does the nucleus
The people we get our
different direction?
Yes.
What you say?
So the nucleus gives different directions for different, you all have it's like having the same library, but you're reading different books, right? We all have, your DNA is your library, but each cell type reads a different book. So when you're making an eye, your eye cells say, okay, we're going to read the eye book, right? And when you're making a brain cell, you're reading the brain book, right? But we're not really talking about books though, are we?
No.
We're talking about these things, genes. So if orange is on, let me go ahead and do this and do on, right? And red is off, right? Shh. Some of your genes, if you're going to make an eye cell, this is the pattern you get. And of course, there's many more cells, but we're going to go, many more genes, but we'll go ahead and say this. So you have these genes are turned on, those genes are turned off. That's the pattern when you're making an eye. When you want to make a red blood cell, then try to make the same number of twists here. You have these on, and then this one ends up turning off. Oops. So your pattern's different. So you're reading different, you're turning different genes what? On and off. That's right, on and off. And what's really, really, really interesting, and by the way, this is directly related to your end of course exam because I promise you, you're going to have some kind of question on this on that end of course exam. Guaranteed to have it on the practice test. I didn't see the actual test, but the kids later said, hey, that was on there. So I'm telling you, understanding that you have all these different cell types, okay, and that how do you get one cell that whose original cell was this, has this, all of these have the same DNA, is that here you have a series that it's which genes are turned on and which genes are turned off. It's really that simple. So you have all of us, your whole body, all 1 trillion cells have the same library. They have the same books. But the difference is that these cells are reading one book, these other cells are reading another book. That's the difference. Are we all good with that? It's just turning different genes on and off, right? And so even though you have the same code in each one, it's which code are you turning on, which code are you turning off. And that's going to give you the different cell types. Now, interestingly, we're not done there, not done yet. I don't understand why this thing doesn't want to. All right, good. So that explains why different genes, why different tissues have different DNA. But something miraculous happened about 10 or 15 years ago.
And you was born without a head.
Yeah. Shh. He's just, don't even listen to him. All right, so something miraculous happened about 10 to 15 years ago is we found out that the gene sequence isn't the only part of the answer. There was this thing called epigenetics. It's actually nothing more than turning genes, different genes, on and off. And by the way, turning a gene off means you get different RNA. Turning a gene on means you get different RNA. So that's why your answer was a great guess. So epigenetics is just this turning different genes on and off. And what we found out is that you can, for instance, if you drink too much, or you don't exercise, or you're obese, or you have diabetes, or your lifestyle, you smoke, you drink, I guess I think I already mentioned drink, or do drugs, those chemicals can turn certain genes on and off. And you can pass that on to your children. Right? You can pass out. So you smoke. The effects of smoking doesn't just increase your chances of cancer, right? It can actually be passed on to your child. And then that child can pass it on to, without that child ever smoked, that child can get the negative impacts of that smoking.
That's how you get AIDS and stuff?
No, let's not go crazy. Now we're just jumping around. AIDS. Infectious diseases are another issue. We're talking about genetic issues, which you don't understand that yet because we haven't gone into it, but viruses and bacteria can invade your body. We call that disease infectious disease. Are you clear?
I have no question.
Now listen carefully. Viruses and bacteria attacking your body, that's an infection. The kinds of diseases we're talking here about, sickle cell anemia, the impacts of smoking, right? That's your genetic makeup, your own DNA. What genes you turn on and off. So it's just very interesting to me that your lifestyle can impact your children moving forward many generations. A few questions, we don't have time for all these questions, so go ahead.
So there could be an epigenetic that would affect if there's like something that gives you like a stroke or something.
Could there be an epigenetic
That turns
event that can turn genes on and off that make you more susceptible to strokes and then give your child more susceptible to strokes and your children's children more susceptible to strokes? The answer to that is yes, that could happen. Yes. Oh, this one here, what kind of question is that? Yeah.
So it's about these genes.
Yeah.
Since there's different cells, like for one cell, let's say you're making the eye, for the certain cell for the eye, would each strand of DNA be different, or would it be the same?
It's a great question. It's what I said earlier. This DNA is the same. The sequence of the DNA, AT, GC, GC, TT, AT, is all the same. The question is, which ones of these genes are turned on and which genes are turned off?
Right.
And that's through chemical signaling. And we'll talk about that in just a second. So if a cell is here, when it's being developed, right? Let's say it's here, and another cell is over here. This one is going to get different chemical signals, different concentrations, right? Because of diffusion and osmosis, you have different concentrations of chemical signals, right? And so this one, because of the chemical signals, turns off genes 1, 13, 206, right? Where this gene gets a different set of signals, it turns off, keeps one on, but turns off 4, 24, and 206. And there's more, right? There's a bunch of differences, but I'm just giving you this as an example. So then this one turns out to be one cell type, and this one turns into another cell type. And we call that embryology. The study of this is called embryology. So we know that chemicals and different chemical concentrations can signal your DNA to turn different genes on and off. But the interesting part that started to develop an understanding, we started to develop an understanding of 10, 15 years ago, is that all these chemicals, all these life decisions that you have, that I have, that we all have, we can send those changes, we can change those ons and offs, can kind of stay semi-permanent. You can turn certain genes on and off, that they stay, that increased risk of stroke, that increased risk of heart attack, that increased risk of diabetes. We can send that to our children and then our children's children through our DNA. So our environment, our actions now impact our DNA and the DNA of our children. That's kind of deep, isn't it? So epigenetics is the study that, again, relatively recently in the study of DNA did we understand this. When Watson and Crick and Rosalind Franklin first started working with DNA and their structure, they were only talking about a sequence of DNA. They were thinking simple, right? They said there's a code, you take the code, you make a protein, the protein makes a cell, the cell, depending on which code you get, you make this, but things are a lot more complicated. Yeah?
Epigenetics. Does that cause like people to have like addictions and stuff to turn yourself? Could,
this is all could, right? Could, I drink a lot. Maybe I'm an alcoholic, maybe I'm not. Could that drinking then make my child more susceptible to drinking?
Yes.
The answer is yes, it could. We don't know. This is all very experimental. There's a lot of stuff that we're learning. We're learning it still. The people are working on it, they're testing it. So there's all kinds of questions. We know that, and we do it through that, remember that methyl group we talked about before earlier in the year, that CH3 group? Well, that's what we do. That's one of the ways you can change the genes. That's how we can turn them off and on. We can add a methyl group to the gene, and we can actually stop it. It's called methylation. There's other ways of doing it, but that's a very kind of really just straightforward way, if in case you're wondering what kind of signal it is. But whether something's methylated or not can be influenced by drugs we take in, by lack of exercise, by exercising, by eating too much, by not eating enough. So all these choices, and what is the impact? Who knows? We don't know yet. With a lot of it, there's a lot of studies being done. There's giving us a lot of information, but we're still kind of working on our understanding of this. Yes?
Okay, so somebody in my family has cerebral degeneracy. So that could have been caused from something that my great-great-great-grandparents...
Yeah, yes, absolutely. It's very hard for people to conceive this. My little brother was born with his feet on backwards. Yeah. And the reason that happens, my mother grew up in the 60s, and they didn't put a connection between cigarette smoke and malformations, right? And my mother gave birth to my brother with his feet on backwards because of smoking's impact, the chemical signals impact on those directions that we just talked about. So that smoke, those chemicals changed the signals. Now, thank God my mother was able to find a surgeon who was able to correct it. And let me tell you, it was the way they corrected it. They broke his little ankles over and over again and kept straightening it out. Kept putting casts for years. They did that, and he ended up with very solid, well-formed feet. He's able to run around, walk around now, and normally he went to the Army, he's an airborne ranger. Just everything, just absolutely normal. But it was through the grace of God and a series of scientific, discoveries that we allowed the surgeon to do what they was able to do, right? And those chemicals, though, initially changed the way the signals, the cells that were making him, right? Because he was being made in my mother's womb. Those chemical signals were impacted by the smoke that my mother took into her lungs. The same thing happens with alcohol. The same thing happens with not exercising. The same thing happens with exercising and with not smoking. So we just don't know what's going on, right? So it's just a lot of questions and answers that need to come. Yeah?
One, if a person has multiple children and they did something to where they messed with the genes, is it sent to both? Or
it's a great question. So when you're thinking about these kinds of changes, here's what you got to think about. If you think of your cell, your sperm, your father's sperm, or your mother's egg, right? When you're taking in chemicals, it's like someone crossing a field with another person shooting at them. So if think of yourself trying to cross a field. Could you cross a field and someone's shooting a bullet at you with a handgun? Could you dodge, zigzag, and make it to the other side of the field? Yeah, it's possible. It's possible. And yeah, you could get shot. You could get hit. I'm hit, right? Or you might make it to the other side. When you're hit, it would be like that idea that those DNA, that chemical hit that cell, changed the DNA. Does that make sense? So now imagine someone's now, instead of just a handgun, somebody shooting a machine gun into that field. Your risk of getting that change in your DNA has what? Increased, right? So your chance of crossing that field safely has decreased, or your chance of getting hit have increased. So it's like that. So when you smoke, like let's say you're at a party, you're pregnant, you take one glass of wine. Well, you just shot at your baby, but probably missed it because it's a big field. But if you go home every night and drink a bottle of gin, or whatever, and you're shooting a machine gun at your kid. Is it possible that kid is born with no problems? Sure.
A little bit of alcohol.
But whatever. But sure that those changes, maybe they miss. But you've increased the risk so much. So what's the risk to smoking, taking drugs, or drinking alcohol? Look, I don't know the numbers. I would suggest that I would put forth to you that not doing it is probably the safest route. In fact, you all need to think about this. It's not just alcohol, it's not just cigarettes, it's not just drugs. Drugs in the sense of illicit drugs. It could be taking too much vitamin A. Too much vitamin A, you would think, vitamin? What? It's a vitamin. Why would it too much cause it? It turns out that too much folic acid, vitamin A, can cause your fetus to have malformations.
What's vitamin A?
Vitamin A is just a vitamin. It's in spinach, it's in different greens. Taking low amounts is necessary. You have to take a decent amount, but taking too much vitamin A could cause problems for your fetus.
No more Thanksgiving.
No, hey, could you try to listen and try to not to process in a joking style? Because there could be someone in this room right now as a little brother or sister that's born with a malformation, and they really don't want to have any kind of laughing about the serious topic. Yes.
Okay, so I got a question.
Yeah.
So, if you're not pregnant, you smoke marijuana and stuff, and then
Say again.
if you're not pregnant, you smoke marijuana and stuff, and later on in life you have a child, can that still affect your brain? 'Cause doesn't marijuana-
So if you're not pregnant and then you do smoke marijuana, so you're smoking, and then you stop, you're having a baby, so you say, I'm not going to smoke because I'm pregnant. You're asking, can that still? Yes, that's the point. Is that when you're smoking marijuana, even though you're not pregnant, that could, now I'm not saying yes or no, right? When I say, you're asking me, could it, I'm saying yes to could, right? Like I'm saying, it's like shooting a machine gun at your DNA. You're taking these chemicals in, those changes, those genes being turned on or off, those chemicals are turning your genes on and off. Including the genes that are in your ovaries, in your eggs.
But what if you stop wait?
I can't tell you, I wish I knew. I don't think anybody knows. I don't think anybody knows the answer to that. I'm just telling you that your lifestyle can impact your children. I'm not trying to put a guilt trip on you before you even have kids, right? I'm just telling you that impossibility is out there, and that you really need to think about how you live your life and your choices you make. The answer to your question is will it do this or will it do that? I can't give you that answer because I'm not even sure if anyone knows. I'm sure there's some information, there's a lot of information out there I don't know, but I don't even know if that exists.
Okay, now I got some.
One last one, gotta get going.
Okay, so this lady, she said that it's actually good to drink like a little bit of wine when you're pregnant.
Here's what I always say the following. I know two things. I know two things. I probably know a few more than a few things, right? But a couple things I do know, and that there are doctors out there with an answer to that. And I also know I'm not that doctor. I'm not a doctor. I don't have that information for you. So I heard that one glass of wine can't hurt you. I have heard that. But number one, I've never heard that one glass of wine will help. So I've never heard that. I have heard one glass, but that's what I heard. What did I say initially? I'm not a doctor. So what my suggestion is, if you're planning on getting pregnant or you are pregnant, you should not take alcohol, is my opinion. If you want to talk, and if you can't go 9 months without drinking alcohol, then maybe you need to revise your identity and maybe think that you might be an alcoholic. Because literally, if you can't go, you know what I'm saying? You can't go 9 months without drinking a drink of alcohol, you have a problem, I think. That's my opinion.
Does he look like me at all?
All right, so I don't drink that much. I go months without drinking. I don't know if people need drinking for whatever social reasons. I think if I had a baby growing in me and there was a risk of that baby,
And you're shooting
I wouldn't shoot at my baby is what I'm saying, right? Okay, finishing, thank you. All right, so we go from one cell when we're conceived. How are we conceived? Your mother released an ovary. That ovary went through the fallopian tube. Your father sent sperm up from the bottom, and finally they met. As soon as they met, you became conceived. Conceived, thank you. You were conceived at that moment, but you're still a single cell.
What?
You can't. I'm sending it to you. Shh. It's just basic human anatomy. It's not a big deal. It's going to be in the video. Please be quiet. It's really not that big a deal. You're not children anymore. You're adolescents. Could we try to behave that way? Enough with the comments. So the sperm meets the egg, it's conceived, you became a human being at that point. A full human being, half the DNA from your mom, half from your dad. It was mixed up during meiosis, you're an individual, you're a very different individual from anyone else in the whole planet. Really amazing moment in your existence. But now you're one single cell, you have to become a trillion cells. And not just a trillion cells, but a trillion cells, many of which have different functions and structures and colors and do just all kinds of complicated steps in your development as a human being. You started off with one cell and you became what was called, this little ball of cells. And then in that ball of cells you have different layers. And depending on where you are in that layer, your different DNA is turned on and off. You have different genes that are turned on and off. And some of those genes, like this outer layer here, can be in this kind of diagram, which is not true. Let me say this right now. There are plenty of diagrams out there that are accurate. This is just a for instance, okay? The outer layer can become your central nervous system or peripheral nervous system, right? Maybe the middle layer becomes your skin. Maybe the inner layer becomes your muscles. Just a for instance. So that's at that ball stage of you being, you didn't go from a single cell and then that cell didn't pop out a little head and 2 little feet and 2 arms, right? That didn't happen. And what happened was that they kept dividing it, each one specialized, each one with a different structure. So we'll move on to this. You need to know this. You need to know this chart. The very first chart here says Totipotent. That means that cells that have the total potential to become you as a whole. Totipotent. Each cell can develop into a new individual. That only exists on day 1 through 3. So on day 1 through 3, if you divide, if those 2 cells separate at days 1 through 3, what do you have?
Twins. Caitlin and Lauren.
Because each one of them can now become a separate human being. But if they divide at days 5 through 14, you can't have twins. Does that make sense? Because those cells are specialized. So at this point, nothing's been turned on or off. They're just dividing. At this point, you're turning genes on and off. But here you've turned genes on and off so that days 5 through 14, you can make things that are, like endoderm, mesoderm, or ectoderm. And those 3 then, once you turn more genes off and on, you can then say these cell types can become lung and pancreas. These can become our muscle and blood, and these can become skin and nerve cells. Before you go, let me say this. Know that viruses are not cells, and what's the difference? And that's it. That's the test. That's what's going to be on the test on Monday. Yeah.
So, it would be 1 to 3 days after being conceived. If they split, you'll have, more than one human being, right?
1 to 3 days. If they split, they can become full human beings.
After being conceived.
After being conceived, that's right. So that's how you get identical twins. They split after they've been conceived. Fraternal twins are when you have more than one egg being fertilized by more than one sperm. Triplets and quadruplets, the egg splits more than once. Like triplets, you have one cell that splits into 2. Now the 2, and then one of these splits again. So now you got 3. In quadruplets, they both split twice. In octuplets, Say what? I can't hear you because these guys are too loud. Say again. Yeah, those are all identical twins. Now, if you're talking fraternal twins, you're talking 2 eggs, 2 separate sperm. Those are fraternal twins.
Sir, those are mine. We get to pick either plant or animal to make.
Take your honeybee
Scratch the whole project because I haven't been able to get all the equipment I wanted to get for you guys. And I'll send you an email to that, in fact. Study for the test, though. I'm going to send you this video. You guys need to review the video. All right?
So it's no homework. You just have to study for the test.
Yeah, just watch this video, which I think is pretty short, but it's probably an hour long. I'm not sure. All right?
Mr. Mendoza.